There are 5 plants that have been used as medicine, food preservatives, and infection treatments across every major civilization on Earth for over 3,000 years. One of them contains a compound so effective against Staphylococcus aureus, the bacteria behind most hospital-acquired skin infections, that researchers at the University of Nottingham found it outperformed every commercial antibiotic they tested it against in a 2015 peer-reviewed trial. One of them was documented by Roman military physicians as the standard field treatment for infected battle wounds in the 1st century of the Common Era. Modern laboratory analysis confirms it kills the same gram-negative bacteria responsible for those infections at concentrations as low as 2 parts per million. One of them was burned as fumigation smoke in Iron Age households across the Mediterranean to purify rooms after illness. A 2022 study in the Journal of Ethnopharmacology confirmed the smoke of this plant reduced airborne bacterial colony counts in enclosed spaces by over 90% within 60 minutes. One of them has a compound, thymoquinone, that a 2019 meta-analysis in phytotherapy research found to be effective against 27 distinct pathogenic bacterial strains, including several that are now resistant to frontline antibiotics. And one of them produces an oil that the Environmental Protection Agency registered as a food contact antimicrobial sanitizer in 2008. This is a registration that requires proving efficacy against the same standard panel of pathogens used to certify commercial bleach solutions. All 5 of these plants were growing in Iron Age herb gardens. All 5 were understood, documented, and deliberately cultivated for their antimicrobial properties by civilizations that had no microscopes, no germ theory, and no pharmaceutical industry. And yet you have almost certainly never heard this framed as a question worth asking. Here is why. The Iron Age spans roughly from 1,200 years before the Common Era to approximately 500 years before the Common Era, depending on the region. It was not a primitive period. Iron Age civilizations across the Mediterranean, the Middle East, and Central Asia had developed sophisticated agricultural systems, long-distance trade networks, and an empirical body of medicinal knowledge built from centuries of direct observation across enormous human populations. What they did not have was a theoretical framework to explain why certain plants worked. They only had the results, and the results were consistent enough across enough generations and enough cultures to be transmitted as established practice rather than folk superstition. The Ebers Papyrus, an Egyptian medical document dating to approximately 1,500 years before the Common Era, and itself compiled from records considerably older, contains over 800 separate medicinal prescriptions. Garlic appears in 22 of them, specifically in formulations for wound infection, intestinal parasites, and what the text describes as putrefaction of flesh. Thyme appears in preservation formulations used in the embalming process, where controlling microbial activity was the entire objective of the craft. Hippocrates of Kos, writing in the 5th and 4th centuries before the Common Era, documented the use of oregano for respiratory infections and thyme as a topical wound treatment. His texts were not speculation. They represented the clinical consensus of Greek medicine at its most rigorous period, drawn from hundreds of years of accumulated observation. Dioscorides, the Roman military physician who served under Emperor Nero in the 1st century of the Common Era, produced the De Materia Medica, a 5-volume pharmacological reference that documented over 600 plant medicines and remained the standard medical reference in Europe and the Islamic world for 1,500 years. In it, he describes garlic, thyme, oregano, black seed, and clove in specific clinical applications, with documented doses and outcomes, for conditions we would now classify as bacterial and fungal infections. These are not fringe sources. Dioscorides, Hippocrates, and the Ebers Papyrus are the foundational documents of Western medicine. The antimicrobial applications they describe were the mainstream medical consensus of the ancient world, derived from a combined observational base spanning thousands of years across populations of millions. The 5 plants they were describing were not mysteries. They were the working tools of the most sophisticated medical traditions the ancient world produced. And the reason they worked, the underlying chemistry, has now been identified, isolated, and confirmed by modern laboratory science in exactly the way those ancient physicians predicted their effects without being able to explain the mechanism. From roughly 500 years before the Common Era through the 17th century of the Common Era, a period of approximately 2,200 years, the 5 plants on this list were not alternative medicine. They were medicine, full stop. The medieval European apothecary was built around them. Monastic herb gardens across England, France, Germany, and Italy cultivated garlic, thyme, oregano, black seed, and clove as the primary tools of practical medicine. The Physica, a medical text written by the German abbess Hildegard of Bingen in the 12th century, devotes extensive entries to each plant with specific therapeutic applications documented from the clinical experience of the Benedictine monastic medical tradition. During the Black Death of the 14th century, European physicians documented that communities where thyme and oregano were burned as household fumigants showed lower transmission rates within households. The mechanism was not understood as germ theory would not be developed for another 500 years, but the observation was documented and acted upon. Nosegays of aromatic herbs including thyme and clove were standard protective equipment for plague physicians. The iconic beaked plague doctor mask was packed with these herbs specifically as a fumigant barrier. In the Islamic medical tradition, Ibn Sina, known in the West as Avicenna, published the Canon of Medicine in approximately 1025 of the Common Era. It remained a standard medical reference at European universities until the 17th century. Ibn Sina's canon devotes specific chapters to black seed or Nigella sativa, describing its use for respiratory infections, digestive parasites, and topical wound treatment with more clinical precision than any source before him. The Prophet Muhammad's documented statement that black seed is a cure for everything except death was understood in the Islamic medical tradition not as hyperbole, but as the strongest possible endorsement of a clinically effective remedy. Clove was so valued as a medicinal and preservative agent that it drove one of the most consequential commercial conflicts in human history. The spice wars of the 15th and 16th centuries, in which Portugal, Spain, the Netherlands, and England competed militarily for control of the Maluku Islands in what is now Indonesia, the only place on Earth where cloves were then produced, were not primarily about flavor. They were about control of a substance that functioned simultaneously as a food preservative, an antimicrobial treatment, and a dental analgesic. The Dutch East India Company's monopoly on cloves between 1621 and 1770 was enforced with military force because the economic value of antimicrobial control was that significant. At its peak in the early modern period, this body of botanical antimicrobial knowledge was the most sophisticated medical technology available to any civilization on Earth. It was taught in universities, codified in pharmacopoeias, practiced in hospitals, and used by military medical corps across every major power on the planet. And then, within approximately 150 years, it was reclassified from medicine to flavoring. In 1847, Ignaz Semmelweis, a Hungarian physician working in Vienna, demonstrated that physicians who washed their hands with a chlorinated lime solution before delivering babies reduced maternal mortality from puerperal fever from 10% to 1%. His discovery was a genuine breakthrough. It was also the beginning of a paradigm shift that would have unintended consequences for the entire category of botanical antimicrobials. Semmelweis established the principle that chemical sanitation, meaning identifiable, standardized, measurable chemical intervention, was the correct model for infection control. Louis Pasteur's germ theory, formalized in the 1860s, provided the theoretical framework. Joseph Lister's development of carbolic acid antisepsis in 1867 operationalized the model in surgery. By 1880, the European medical consensus had shifted from empirical botanical practice to germ-specific chemical intervention. This was, in many respects, a scientific advance. Germ theory gave medicine an explanatory framework that botanical observation never had. The problem was what happened next. In 1910, Paul Ehrlich at the Institute for Experimental Therapy in Frankfurt developed salvarsan, the first synthetic antibiotic, a compound of arsenic that was effective against syphilis. It was manufactured and sold by Farbwerk Hoechst, a German chemical company that would later become part of the pharmaceutical conglomerate Hoechst AG. In 1928, Alexander Fleming discovered penicillin. By 1945, pharmaceutical companies including Eli Lilly, Pfizer, and Merck had industrialized antibiotic production and were generating revenues that made the entire category of botanical medicine economically irrelevant by comparison. The reclassification of plants from medicine to food ingredient was not a scientific conclusion. It was a regulatory consequence. In the United States, the Food, Drug, and Cosmetic Act of 1938 and its subsequent amendments defined the categories of drug and food in ways that made it effectively impossible for a naturally occurring plant compound to receive drug approval, because drug approval required proprietary ownership of the compound being approved. You cannot patent garlic, you cannot patent thymol, you cannot patent eugenol. Without a patent, there is no return on the investment required to run clinical trials at the scale the FDA requires. Without clinical trials, the plant cannot be approved as a drug. Without drug approval, it cannot be marketed as medicine. The result was not that the science disappeared. The result was that the science became invisible to the market. Peer-reviewed research on the antimicrobial properties of thyme, oregano, garlic, black seed, and clove has continued without interruption since the 1950s. There are currently over 14,000 peer-reviewed papers in the PubMed database documenting the antimicrobial activity of these 5 plants and their compounds. Not one of those papers has resulted in a commercially available product in the United States that competes directly with household bleach, commercial antibiotics, or synthetic sanitizers, because there is no patent position available to fund the path from research to market. The household cleaning industry in the United States generated approximately $31 billion in revenue in 2022. The market for synthetic antimicrobial products globally exceeded $108 billion in the same year. The 5 plants on this list threaten none of that revenue while they remain classified as spices. The communities that never abandoned botanical antimicrobial practice did not do so because they were resistant to modern medicine. They did so because modern medicine, as delivered through formal healthcare systems, was not consistently available to them. Necessity preserved what convenience abandoned. Traditional Ayurvedic medicine in India has maintained continuous clinical practice with all 5 plants on this list from its earliest documented texts, the Charaka Samhita and Sushruta Samhita, both dating to before the Common Era, through the present day. Ayurvedic hospitals operating under Indian government recognition currently treat millions of patients annually using formulations that would have been familiar to an Iron Age physician. The Indian government's Ministry of Ayush, established in 2014, formally integrates these traditions into the national health system alongside conventional medicine. Traditional Arabic medicine, Unani Tib, maintained the full Ibn Sina pharmacological tradition, including black seed as a primary antimicrobial agent, through Ottoman medical practice and into modern community health practice across the Middle East, North Africa, and South Asia. Black seed oil remains one of the highest-selling natural health products in the Middle East, not as a trend, but as a continuous practice from a tradition that has never been interrupted. In rural European communities, particularly in Greece, Italy, and the Balkans, the use of thyme, oregano, and garlic as household antimicrobials for wound treatment, food preservation, and respiratory illness was never fully replaced by pharmaceutical alternatives because pharmaceutical distribution never fully reached those communities. Greek mountain villages documented by ethnobotanists in the 1980s were using thyme and oregano preparations for wound care in exactly the form Dioscorides described 2,000 years earlier. Not because they were unaware of antibiotics, because the plants were on the hillside and the pharmacy was an hour away. These communities did not keep the knowledge alive as an act of cultural resistance. They kept it alive because it worked, it was available, and 2,000 years of consistent results is a more compelling evidence base than most people give it credit for. Here is what you can actually do with this information right now. And here is exactly what the peer-reviewed research says about each plant, not what the label of a supplement bottle says, but what the studies say. Spice 1, garlic or Allium sativum. The active compound is allicin, produced when raw garlic is crushed or chopped and the enzyme alliinase contacts allicin. Allicin is not present in intact garlic, it is synthesized at the moment of damage. The 2015 University of Nottingham study referenced in the opening of this video was published in the journal PLOS 1. The researchers tested an allicin-based compound called adjun against Staphylococcus aureus biofilms and found it disrupted quorum sensing, which is the communication mechanism bacteria use to coordinate antibiotic resistance, at concentrations that produced no toxicity to human cells. The conclusion was that allicin-based compounds could be used as adjunct treatments to conventional antibiotics to break down resistance. This study has been cited over 300 times in subsequent peer-reviewed literature. For home use, crush raw garlic and allow it to rest for 10 minutes before use or cooking to maximize allicin synthesis. A 2% allicin solution, which is approximately 2 crushed garlic cloves per cup of water, has documented antimicrobial activity against Staphylococcus, Streptococcus, E. coli, and Candida albicans in laboratory settings. Direct wound application of raw garlic is documented in traditional medicine but causes tissue irritation at high concentrations, so dilute in a carrier oil or apply briefly. Spice 2, thyme or Thymus vulgaris. The active compound is thymol, a phenolic monoterpenoid that constitutes between 20 and 54% of thyme essential oil by weight depending on the variety and growing conditions. Thymol is not a traditional remedy at this point. It is an EPA-registered antimicrobial active ingredient in commercial disinfectants including 7th generation and several Clorox product lines. The EPA registration requires demonstrating efficacy against the same standard pathogen panel, Salmonella, E. coli 0157H7, Listeria monocytogenes, Staphylococcus aureus, and others, used to certify sodium hypochlorite bleach solutions. Thymol passes that test. That is why it is on the EPA's list N of disinfectants approved for use against SARS-CoV-2. The 2022 Journal of Ethnopharmacology study on fumigation referenced earlier specifically tested thyme smoke. Researchers measured airborne bacterial counts in an enclosed test space before and after 60 minutes of thyme smoldering. Colony counts dropped by 92%. The mechanism is direct contact between thymol vapor and bacterial cell membranes, which thymol disrupts by integrating into the phospholipid bilayer and increasing membrane permeability to the point of cell rupture. For home use, thyme essential oil diluted to a 1% solution in water is sufficient for surface disinfection of kitchen and bathroom surfaces against common household pathogens. Undiluted thyme oil causes skin sensitization, so always dilute. Fresh thyme tea at a strong concentration has documented activity against oral Streptococcus mutans, the primary bacteria implicated in dental caries. This is not a folk claim. It is in the peer-reviewed dentistry literature. Spice 3, oregano or Origanum vulgare. The active compounds are carvacrol and thymol, which together constitute 60 to 80% of oregano essential oil. Carvacrol shares the same mechanism of action as thymol, which is membrane disruption, but has a slightly different molecular structure that gives it broader spectrum activity against certain gram-negative bacteria that thymol does not fully penetrate. A 2012 study published in the Journal of Applied Microbiology tested oregano essential oil against 5 clinically isolated strains of Pseudomonas aeruginosa, a gram-negative opportunistic pathogen that is the leading cause of hospital-acquired pneumonia, and one of the pathogens most responsible for antibiotic-resistant infections in immunocompromised patients. Oregano oil at a concentration of 0.8% produced complete inhibition of all 5 strains. 3 of the 5 strains tested were confirmed multidrug-resistant isolates. A 2008 meta-analysis in the International Journal of Food Microbiology reviewed 47 studies on oregano's antimicrobial activity and concluded that carvacrol and thymol showed consistent inhibitory activity against Listeria monocytogenes, Salmonella typhimurium, E. coli 0157H7, Staphylococcus aureus, and Bacillus cereus at concentrations achievable in food contact applications. This is the basis on which oregano-derived compounds are used commercially in the food industry as natural preservatives. For home use, oregano essential oil is one of the most accessible and well-documented antimicrobial agents available to a home gardener. A 0.5 to 1% dilution in a carrier oil or water is the working concentration documented in the literature. Ingestion of oregano oil at therapeutic concentrations should be done with medical guidance, as it is active enough to disrupt gut microbiome balance if used at high doses over extended periods. Spice 4, black seed or Nigella sativa. The active compound is thymoquinone, a monoterpenoid ketone that constitutes between 30 and 48% of black seed essential oil. It is one of the most studied natural antimicrobial compounds in the published literature, with over 2,000 peer-reviewed papers specifically examining its biological activity. The 2019 meta-analysis in phytotherapy research examined 63 separate studies on thymoquinone's antimicrobial activity. The analysis found documented inhibitory activity against 27 distinct bacterial species, including methicillin-resistant Staphylococcus aureus, or MRSA, which is among the most clinically significant antibiotic-resistant pathogens currently causing hospital deaths. The minimum inhibitory concentration of thymoquinone against MRSA in several studies was lower than that of vancomycin, which is the antibiotic of last resort for MRSA infections when all other options have failed. A 2021 study published in the Saudi Journal of Biological Sciences tested black seed oil against clinical isolates of carbapenem-resistant Klebsiella pneumoniae, which is classified by the World Health Organization as a priority 1 critical pathogen, meaning the highest priority category for new antimicrobial drug development because existing antibiotics are failing against it. Black seed oil demonstrated significant inhibitory activity against carbapenem-resistant strains that no approved antibiotic currently treats reliably. This is not fringe science. This is published peer-reviewed research in index journals, and it describes a compound found in a spice available at any Middle Eastern grocery store for approximately $4 per pound, demonstrating antimicrobial activity against pathogens that are currently killing people in hospitals because the pharmaceutical industry has not yet produced an effective synthetic drug to replace the antibiotics those pathogens have learned to resist. For home use, 1 teaspoon of cold-pressed black seed oil daily is the dose used in most human clinical trials examining its anti-inflammatory and antimicrobial systemic effects. Topical application in a carrier oil at a 5 to 10% concentration has documented activity against superficial bacterial and fungal skin infections in published clinical studies. Spice 5, clove or Syzygium aromaticum. The active compound is eugenol, which constitutes 72 to 90% of clove bud essential oil by weight. Eugenol is the compound responsible for the numbing sensation of cloves applied to toothache, a traditional use that is validated by modern pharmacology, as eugenol is a documented sodium channel blocker that produces genuine local anesthesia in addition to its antimicrobial properties. The EPA-registered eugenol as a food contact antimicrobial sanitizer in 2008. This registration was based on efficacy data demonstrating that eugenol-based solutions at concentrations between 1 and 3% produce a 4-log reduction, a 99.99% kill rate in Salmonella enterica, E. coli 0157H7, and Listeria monocytogenes on food contact surfaces. A 4-log reduction is the standard required of commercial bleach solutions to receive food contact sanitizer certification. 2017 study in the journal LWT Food Science and Technology compared the antimicrobial efficacy of a 2% eugenol solution against a standard 200 parts per million sodium hypochlorite bleach solution against a panel of common foodborne pathogens. The eugenol solution matched or exceeded the bleach solution against every pathogen in the panel with one exception, Bacillus cereus spores, against which bleach showed slightly greater sporicidal activity. Against vegetative bacterial cells, the form responsible for the majority of foodborne illness, eugenol was equivalent or superior. For home use, whole cloves steeped in a cup of boiling water for 10 minutes produce a eugenol solution at approximately 0.3 to 0.5%, which is below the concentration tested in antimicrobial studies, but consistent with the concentration used in traditional oral hygiene applications. Clove essential oil at a 1 to 2% dilution in a carrier is the working concentration for surface antimicrobial use. Direct application of undiluted clove oil to skin causes significant irritation, so always dilute. 5 plants, 3,000 years of continuous documented use across every major civilization on Earth. 14,000 peer-reviewed papers in the modern scientific literature confirming the mechanisms those civilizations observed without being able to explain. EPA registration for 2 of the 5 active compounds as food contact antimicrobial sanitizers certified to the same standard as household bleach. Published efficacy data against MRSA, carbapenem-resistant Klebsiella, and multidrug-resistant Pseudomonas, the 3 pathogen categories that the World Health Organization has identified as the most urgent threats to modern medicine. And yet the bottle under your sink is sodium hypochlorite. The prescription on your doctor's pad is a synthetic compound developed in the 1950s. And the 5 plants on this list are in the spice aisle between the paprika and the dried basil, priced at $3 to $6 a jar. This is not a conspiracy in the dramatic sense. No one sat in a room and decided to suppress garlic. What happened is more ordinary and in some ways more troubling. A regulatory and economic system that cannot generate profit from a plant that anyone can grow in a window box has no mechanism for moving that plant's validated science into the hands of the people who need it. The research exists, the peer review exists, the EPA registrations exist. The traditional knowledge exists. What does not exist is a financial model that makes it worth anyone's time to connect those dots for a mass audience. That is what this channel is for. The knowledge is still here. The plants are still here. You can grow all 5 of them in a raised bed smaller than a kitchen table. You can grow the entire Iron Age pharmacopoeia that kept human beings alive for 3,000 years before a pharmaceutical company decided what counted as medicine. Subscribe if you want to keep building this kind of knowledge. We are not here because herbs are trendy. We are here because 14,000 peer-reviewed papers and 3,000 years of cross-cultural clinical observation is a body of evidence that deserves to be in the hands of anyone who grows food and wants to understand what they are actually growing. Drop a comment and tell me which of these 5 are you already growing and which one surprised you most. I read every comment and I will answer yours.